All right, so I'm trying to finish up here, trying to convince you why metabolism is extremely important. And yes, it's complicated. I really have tried to simplify it as much as possible. And my priorities are such that it is important to understand why oxygen is so vital, that it is that final electron acceptor. Without it, ATP cannot be produced. Without ATP, we will not be living. Our bodies definitely rely on chemical reactions, just like your bodies are composed of cells. All of those cells do rely on chemical reactions. Some are building reactions, some are decomposition reactions, but it does remind me a lot of cooking. So I say that because recipes usually require multiple steps that do have to be performed in a particular order. Many of them will require specific ingredients and tools to get to a final end product. So to get to your final end product. Well, that's very similar to metabolism. A lot of times there will be multiple steps with specific substrates and tools, which are enzymes and coenzymes, that will end up in a final end product or a few end products. And using the concept that you can see those multiple steps, you can start to make predictions on, well, what if I am missing a specific ingredient, or what if I am missing a specific tool, or what if one of my enzymes is damaged? Can you predict what is going to be a consequence of that? So I'm pretty sure you can cook in the kitchen and use your favorite recipe and make your predictions of if-then. If you're making chocolate chip cookies and you forget chocolate chips, guess what? You're not going to have your proper end product, but hey, you might still have some end product that you're happy with. I'd be happy with some cookies that didn't have any chocolate chips. However, let's say you did not have a bowl as a tool to make those cookies, then the chances of your cookies getting made will be much decreased, because trying to make those cookies without some of those tools is almost impossible. All right, so once you have that idea of multiple steps, like a recipe requiring different substrates and enzymes, you can apply that to many of the ways that drugs that are prescribed in America work. Many drugs work to modulate these chemical reactions, because the drug is a chemical. And here I have a series of chemical reactions. Each chemical reaction will be catalyzed by a particular enzyme for this reaction, enzyme for this reaction, and onward to the final product, showing cholesterol as the final end product of this pathway. Those nutrients that we do metabolize or ingest, we will see many of those nutrients can be broken down into pyruvic acid by way of glycolysis, and then some of those will be converted to acetyl-CoA. That's that intermediate or entry step into the Krebs cycle. But we also get acetyl-CoA when we break down fats by way of beta oxidation into 2 carbon bits. That's that beta oxidation route. And these 2 carbon bits can be bound to coenzyme A, and those 2 carbons can then be put together to synthesize more lipid-like structures or fatty acids. Cholesterol would require multiple enzymes working with that initial substrate. Well, what if you did have an enzyme issue? Is it possible to then try to find a drug that is going to bind to one of these enzymes? And if it can bind to that enzyme, such as a statin drug may, can you see how that statin drug is going to prevent the synthesis of cholesterol? And that's why statin drugs are cholesterol-lowering drugs. Now, here is where metabolism and genetics intersect. We did say that enzymes are composed of proteins. Enzymes equal proteins. Where does the information come from to make those proteins? Well, it's contained in our DNA. So we have seen in genetics that information is stored in our DNA, and then it will be expressed by having a complementary copy of that DNA transcribed into RNA. That RNA will be translated to produce a protein. I did mention enzymes are proteins. They will have their specific three-dimensional shape, and we'll have that primary, secondary, tertiary shapes. We don't have identical genes for all of us. Yes, we do share some similarities. However, there may be some differences that can promote the production of slightly different proteins in all of us. So we may have very slightly different shaped enzymes. Remember, I think of enzymes like this. However, we might have some modification of a tool where our tool is slightly different in shape, but it might still have a very similar function. Then, when we think about drugs used to modulate these pathways, can you imagine that one statin drug is going to potentially bind to patient A's enzyme that leads to the production of cholesterol? Maybe that drug is going to bind to it extremely well. I'm just going to use the eraser as my statin drug. Here's my enzyme that's going to lead to the production or building of cholesterol. The statin drug may completely bind to that enzyme, and now that enzyme is inhibited. However, what if patient B has an enzyme that's utilized in that cholesterol production pathway? However, it just has a slightly different shape. There might just be a couple of amino acids that are different, and if that slightly different shape has that statin drug not binding as it should, can you see that that particular statin drug is not going to be effective for patient B or patient 2? So a drug that may bind perfectly to patient 1's enzyme and reduce cholesterol may not bind to person 2's enzyme to reduce cholesterol. So that's really the underlying reason why many drugs may work on one patient perfectly, but not on another patient. Wouldn't it be nice to know in advance what drug is going to work best for that patient, rather than just having them be the test tube and trying out different drugs? This is going to be the future of personalized medicine. Is it possible for us to genetically sequence that person's enzymes? Yes, we do have the ability to look for specific gene sequences in people now. And we can also then predict, okay, with that gene sequence, we know that that enzyme is going to be in this configuration, and statin drug number 1 is the best fit for that particular shaped enzyme, versus patient 2 that has a slightly different shaped enzyme. That's where a second or third type of statin drug may work best. But being able to genetically screen those patients in advance will lead to the personalized treatment that will suit them best and be most effective. So it is the future of medicine, that personalized medicine. And personalized meaning, yes, we do share enzymes, but sometimes those enzymes will be slightly different shapes. We can predict what shaped enzyme we all have by looking at our gene sequences. All right, that's the segue into genetics.